Scientists on both sides of the Atlantic said today they'd completed a rough draft of the entire human genetic code.
A revolution in medical science whose implications far surpass even the discovery of antibiotics.
In coming years, doctors increasingly will be able to cure diseases like Alzheimer's, Parkinson's, diabetes, and cancer.
Hello and welcome to Talk Chasers. Welcome to a very exciting video which involves all of these people. Thomas, you might know.
Hi.
From work. Russell, you won't know, but he's here. Christopher from Urban Automotive. Hello. And Tom from Urban Automotive. We are at the Cancer Research UK Care Base, Science, Technology Base, basically where they do all the experiments and stuff on how to cure cancer and stuff like that. Obviously, you might know by Instagram, we're going on a Crush Cancer Not Cars trip. Follow the Instagram on Instagram, and donating pages will be in the link in the description. But we're going on a 3,000 mile round trip to Romania.
Yep.
To raise Transfăgărășan Highway. Transfăgărășan Highway. To raise money in aid of cancer research. Last year the guys went on a tour. Maybe you guys can talk a little bit more about the tour.
Last year we did £500 cars, 500 to spend on the cars. We kept it all a secret. We raised 20 grand in 4 days, but we did 1,700 miles. So this year, to up the ante, to help raise more money, we're doing 3,100 miles in 5 days in £500 cars still. So I think the first day is 810 miles to Vienna. Then to Romania the following day, 790 miles, then to Budapest, then to Nuremberg, then to home. So we've got a lot of miles to do.
However,
£500 cars.
They're not just £500 cars, are they?
90s British touring cars.
So last year there was a theme of just kind of modify.
Yeah, just hide them from everyone else. That was kind of the thing. This year we knew we needed to up the ante to raise more money, and we needed to do more to ask people to dig deep.
Yeah, make it more difficult.
Again, so we're challenging not just the cars but ourselves too. So that, Cancer Research UK, not just us, but we got an invite because of the work we did last year to come along to see where the money goes, so where you guys put your money, how it works. So I don't know if we're going to be allowed. There's a bit of a question mark, but some of the scientists we might be able to speak to. We've got a guided tour of the facility of what goes on, and then some seminars later on. So
it's going to be exciting. If we are allowed to film, it'll be amazing.
That'd be cool.
Doesn't matter, we'll talk about it afterwards.
But yeah, we can talk about it at the end. So
that's going to be today's video. If you're not already part of the Talk Nation, make sure you subscribe. Make sure you're following the link into the description to go and donate to our Crush Cancer Not Cars page. There's different teams. There's Team Ginger Nuts, there's Team Stance.
But also just make sure you donate to the Crush Cancer Not Cars rather than Cancer Research UK. It all gets in the same pot, but we're trying to raise a
exactly. So the link will be in the description that you can click and donate some money to. And let's hope we can crack on with the video and film some cancer research stuff. Oh. I should pre-warn you, a lot of the videos will have like a language warning. I'm going to do like a little intro language warning because I know you can't keep your mouth shut. Yourself.
The Wellcome Genome Campus is where the first animal genome of a simple nematode worm was sequenced in the late 1990s. And now, the campus is leading a revolution in genomics and biodata, bringing the prospect of previously unimagined medical breakthroughs.
Okay, so we know that that's correct, don't we? Being a
Do you reckon that's right?
Oh, we're going to get another...
I think alcohol's before diet.
Sorry, I didn't miss the actual question.
In terms of
the risk factors.
I think
so we know that.
I think sounds pretty.
The most risky in terms of what's going to lead on to. Okay.
Yeah.
I think so.
Do you think sun should be swapped with a drink?
Yeah. I think drink should be further up.
I think no, not sun with drink. I mean drink with food.
But I think sun's higher because not everybody would drink.
Okay, carry on like that.
Carry on like that, and
Carry
we'll
on
see.
like that. Yeah. Okay. Keeping a healthy weight.
Jesus.
Actually, and we know that it is predicted in the next few years to actually start to overtake cancer as one of the smoking as one of the biggest risk factors. Yeah. So number 3 is eating a healthy diet, and as I say, obviously that's closely linked with keeping a healthy weight. But this is about, don't have your bacon sandwiches every single morning. Just have it once a week or something.
That definitely isn't
that processed red meats.
Yeah.
So,
I don't eat red meat now.
So 4 sun then.
So number 4 is sun.
Right.
Yeah.
Okay.
And then you've got these 2 correct. Drink less
weren't too far
alcohol
off.
and being more active.
just saying that they do a lot of research on clinical trials on sort of domestic animals, because domestic animals is naturally not naturally occurring.
It's the same environment we're in.
So rather than making it
Making
it happen where you can't kind of replicate the natural cancer that we get, I suppose, but if the animals have got cancer already, it's a more natural way of testing rather than
artificially induced.
Yeah.
Then that has the advantage that you just want to study one gene, you can just study the effect of that gene because you're controlling everything else. You can't really do that because the dog's going to eat something you didn't know what's going to eat, and that could have an effect. Whereas here,
you know what
we know what we gave it. We know what bedding it's had. It gets changed at a certain time.
We know its whole life story, like its heritage,
and like if you want to study dogs, right? That's population. They're all different. Whereas these mice here, if they're brother sister matings, they're identical. So then you can have a big population of identical things to study, because you can't just study one thing because it could be a weird aberration. You know what I mean?
Yeah.
So there's just two different methods, and then these ones are wild animals.
Yeah, they're like a whole other thing which we looked into, because similar to companion animals, obviously at zoos and also in the wild, pathologists, they will also do reports if they have any animals come in, and they also get cancer in the wild as well. And there are some really interesting examples, like in St. Lawrence in Canada, there's an estuary that was really heavily polluted in the 70s and 80s, and there was a population of beluga whales there that started to develop cancers directly related to the pollutants. And then you saw the same cancers by the same pollutants in the human population happening in the same area. So again, you're getting like the same cancers are occurring, and the same issues and the same kind of factors.
Yeah, so that told you that something in that water was probably the cause
of it.
Yeah.
And then there's the tumours, you can study the tumours. The whales bleached, they died.
And then you're looking at what was in that water, I take it, to find out what it was that was causing that.
And now that's what they found. There is now a known carcinogen. Now we know not to pollute the water.
Who'd have thought?
Also in the wild, on the other end of the spectrum, is you also have natural cancer resistance. So naked mole rats, as far as we know, they don't spontaneously get cancer in the wild. There's only ever been one case which occurred at the Animal Kingdom in Disneyland, and that was because it came from two parents that were bred incorrectly.
Probably stress.
It was probably stress and that kind of thing. But in the wild, there's never been an occurrence of a naked mole rat ever having cancer. And similarly with elephants, despite the fact that they live for a really long time, because that's one of the things that we think is humans live for a long time, we have a long life to accumulate all sorts of things that are going to cause us cancer. Elephants live about as long as we do, but they don't get cancer either, and it's because they have 20 times the amount of copies of a certain tumour suppressant gene than what we have in humans. Yeah,
that's incredible.
So again, it's like interesting to see these resistances in cancer, and we wrote a literature review, we even found cancer in dinosaurs as well. So again, this tells you about cancer evolution, where it's come from, how long it's been around, the different species it affects, all the similarities that can definitely help the human population as well.
Right.
Crazy.
I need to move around like an elephant then.
No, you look like a naked mole rat.
So basically our question is, can we find a new target that will target only cancer cells and not normal cells? The aim of this would be to avoid side effects in therapy and make therapy more beneficial. So basically this whole idea is called synthetic lethality, and the idea is that each of your cells contains multiple genes, and some of the genes can actually perform similar functions. So during evolution, there's basically multiple ways in which sorry. So basically we want to try and find these two genes that perform similar functions, because in a normal cell, if we deleted one of these genes, the cell would be fine because it could rely on the second gene here. Right. The same if we deleted the second gene here, the cell would be totally fine because it can rely on this pathway here because they do the same sort of thing. But if you delete both of them together, the cell will die because it's not able to perform its proper functions anymore. But cancer cells, the reason they're cancer cells is they've already got many changes to their genes. Loads of genes are already lost, they're deleted, they're mutated, they're changed, and that's what's causing the cancer. So our idea here is that because these already have some of the genes lost, can we find this one gene? Because if we can target this one gene, the normal cell will be able to survive because it can rely on the second gene here, but the cancer cell, it doesn't have that second gene, it's already been deleted, so the cancer cell would die. So this is the kind of thing that we're trying to find. There are a few examples of this that have been successfully used in the clinic, but we're trying to find new examples. And we're doing this by using a technique called CRISPR screening. And basically what this allows us to do is it allows us to delete one gene at once in a cell. And then we can look and see if the cell can survive. So when we delete that cell, we delete that gene, does the cell function normally or does it die? And we can do this on a big scale, and actually it's a very big scale because obviously we don't have 2 genes in a human cell. We've got about 20,000 genes in the human cell.
In one cell?
Yeah. So we delete each of them individually. So we have at least 20,000
times?
Yeah, so we've got 20,000 cells, each with one different gene deleted. But we don't just have that because we want to make sure that it's actually working. So we delete each of these 20,000 genes in 8 different ways, using 8 different sequences. And we want to really, really make sure it's working. So we do each of that at least 1,000 times.
You need a lot of scissors, isn't it? Like a lot of snip.
And you're worried about the camera.
So, scientists from all over the world are attracted to work here. Let's see how they define what a genome is.
I would say the genome is a blueprint of all the information needed to generate a whole organism.
Every organism has its own genome. Yeah. Just as the genome connects us with all other humans. You know, we all share much more of our code than we have that is different.
Not only between each species is the genome different, but actually each individual of that species has a slightly different genome.
It's those instructions that define how the natural world looks.
That is much better than I can explain.
So, every living thing has a genome, a code carried in each cell, spelled out in DNA. The human genome is made up of 3.2 billion letters. If printed out, these would stretch from here to the Sahara Desert. From person to person, our genomes differ by just 0.5%. A tiny fraction, which still amounts to millions of differences. These vast data sets have fueled a whole new branch of computer-driven biology called bioinformatics.
And you have an entire floor that's dedicated to just petabytes and petabytes of servers and storage. You know what you're dealing with.
So we're talking about storage for an ever-increasing archive of genomes, with the speed of growth relentless. Just think, it took 13 years to sequence the first human genome. Now, machines in our sequencing labs produce the equivalent of a human genome every half hour, enabling biology on a scale like nowhere else. Large-scale DNA sequencing is also shedding light on a disease that affects 1 in 3 of us. Routine sequencing of tumours is completely changing how we'll diagnose and treat cancer.
In a cancer genome, we see about 10,000 mutations, but maybe only 1 to 10 of those are causal for the cancer.
But by identifying these key mutations, you can start to tailor clinical interventions that have the best chance of outcome for these patients.
So we didn't know that there are different types of breast cancer and that they have to be treated in a different way. And now we know that, and we can also use genomics to inform us about how to treat the cancer.
Genomics is changing our definition of cancer, just as in the same way that genomics is changing our definition of many different diseases.
Scientists here are using the most advanced technologies available to deliver results on an unprecedented scale.
So all of these machines here are DNA sequencers. So you see the ones there that are flashing blue, that's because they're actively reading DNA as we speak. So the screens next to them are sort of flashing up with what the DNA letters are they're reading. So these ones here on my right are the super fancy ones. These ones here are the super fancy ones. So you said in the video about how it took 13 years to sequence the first human genome, and now we can do the equivalent of a human genome in half an hour. This is why, because we've got one of the largest amounts of these kind of sequencing machines, definitely in the UK. We've got some here, we've got some down there as well. So this is to give you a sense of the scale of the amount of genomics and the amount of data that we are producing and analysing here on the campus.
Where you have to use radioactivity to produce the DNAs one by one. When I was in university, I think they just phased this out because it was too dangerous. They replaced it with this. This one is a gel. You have to produce like jellies in between two glass panels. Make sure the glass has no speck or whatever. You have to clean it, otherwise the results come out will get destroyed. And then I think you can do 20 lanes per run.
Is there lines in that? Is it?
Yeah. Once you have the gels, you have a comb. You put a comb inside, and once you take away the comb, you have wells, and you put the DNA in.
Oh.
You got about 400 base pairs. It will take 13 years. That's why to get one genome. We're relying on this sort of technology back then. So this graduates to this.
Yeah.
This one they uses chemicals that produce some fluorescent color. This one uses radioactivity. It's way more dangerous. You have to wear.
So is it those lines you read?
Yes. A, C, G, T. Those are the.
Oh, okay.
So this one graduated to this, and then this one graduated to that sort of stuff.
Right.
And that is our sort of current workhorse. These are the latest and greatest. This is one generation older.
So that's nuts. We've just been in like a little, well, it wasn't, we saw some of the laboratory side of stuff and talk. There's all those books on the genome. That's mapping out the original genome. We've got 3.2 billion cells in our body, and all of those books were mapping out half a cell.
It's just crazy.
Half
and the
a
writing,
cell.
the writing was like,
yeah, you had to use the magnifying glass to read it. It's half a cell, and there's 3.2 billion in the body. It's nuts.
And that half a cell, one cell is made up of half of your father and half of your mother.
Yeah.
So that was just like a mother's
I've got 3
half a
dads.
cell. You got 3 dads. I don't know how that works.
Do you reckon if they got you early enough, they could have found out how to keep you from going bald?
Yes.
So if you look back in the 70s, survival was about 1 in 4 of all cancers that were diagnosed. Back in 2010, 2014, we were about 1 in 2. Which is a great improvement. Over 40 years, we've kind of doubled the survival rate, but we don't think that's quite good enough. And our strategy has been to accelerate that rate of research and that rate of progress so that we can get to 3 in 4 people surviving their disease by 2034. So to do that, that's a lot of research we need to fund so that we can get there in that time. The reason we do this is because cancer is a huge problem. You've got about 360,000 people diagnosed every year in the UK, and that's a huge number of people. And it's also a very complex disease, which is why we need to do all this research, because it's not just, although we give it the same name, it's actually 200 different kinds of disease. This is because we have lots of different kinds of cells in our body, and all these cells have the potential of becoming cancerous. And the same way that your hair and your skin and your blood cells all are very different and have very different functions, when they turn into cancer, they also work very differently. So to put that number into context of 360,000, it's actually estimated that 1 in 2 people will be diagnosed with cancer in their lifetime, which is obviously quite a scary number, especially if you look around the room like this one. But as I said earlier, survival rate has improved massively over the last 40 years, which is great. And this is all thanks to research. And to put this into context, obviously I said we fund a lot. Last year we spent £423 million on research, which is a huge amount of money. And actually we spent 53 million of that in East Anglia, here in Cambridge. Cambridge is a very important place for Cancer Research UK in terms of the work. We have a CIK Cambridge Institute, which is based at Addenbrooke's, but then we also fund people in lots of other locations all across the city. So obviously there's this lovely campus, we fund people at the Sanger Institute and at the UBI, some of them are here today, but we also fund people over at the Gurdon Institute, over at the hospital, and all of these people are kind of working together, communicating and trying to bring forward the day that we beat cancer.
It's basically 3 billion letters that is what makes us human, essentially. And you just need a handful of mutations in these 3 billion letters, a handful of changes to convert a normal cell into a cancer cell. So it's been difficult to study what these key changes are until the development of genome sequencing. So what we can do now is you can take, this is a drawing of a person, you can take a blood sample from that person, you can take a sample from the tumor, and you can compare the two and identify which changes are specific to the cancer, so which changes have led that cell to become a tumor.
And so a story that we published a few years ago, which was using some of the large-scale skills that we have on site, was actually identifying novel, possible anti-metastatic targets to take into the clinic. So metastasis is actually the spread of cancer around your body, and there's a very important critical step which is called metastatic colonization. So this is where the circulating tumour cells have seeded from your primary tumour to a different site, and then they can either remain as they are, they can become dormant because the new environment is hostile to them, or they can continue to outgrow.
Gnomes, garden gnomes, what are we here for?
How's it pronounced?
Genomes.
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 It's like a gangster gnome, you know.
Yeah, it's not genomes.
Like my
genomes, yeah.
Like homes.
And that is made up of thousands of...
If he had a theme tune, it'd be like da-na-na.
So how do you think it is?
Genome. Genome.
What do you think it is?
Genome. What do you want to say, genome?
What do you want to say?
No.
What do you think it is? Come on.
No, okay.
What do you think it is?
Genome.
What do you want to put out there?
Genome.
You think it's a genome, don't you?
What do you think it is? Come on, T-Rex.
I'm not saying anybody's wrong.
What do you think it is?
Well, Ollie.
What do you think it is?
No, I'm not saying you're correct. I think you are correct. But the way I looked at it on the screen earlier, I wonder about...
You've asked me, I've told you what I think. How do you think it's said?
Yes, like you.
So what do you want to go and ask for?
Genome.
Sounds like Chino.
Are we all born, a lot of people ask the question, are we all born with cancerous cells that they need to be activated as such? Yeah, is that the easiest way?
No. So it's the fact they mutate, they're mutated cells. Right.
So that is how adult cancer usually arrives. That is from mutations that are accumulated over the lifetime, which might be speeded up by environmental causes, for example smoking. But it generally, we believe, cancer researchers believe that it is the accumulation of mutations. Whereas in some pediatric cancers, children might be born with a predisposition for cancer. That's why they develop cancer very early on in life.
Okay.
So they might have vulnerabilities in their genome that will drive the cancer very early on in life. So you can think of them as being born either with the
predisposition of
mutations.
Okay, gotcha.
Okay.
Genome as well, by the way.
It's genome.
Yeah, how do you say genome?
Genome?
Genome. Yeah.
No, it's not.
But I'm not a native speaker. Don't listen to me.
No, but
we're having a debate about it. That's fine. So what have we got going on here then?
Oh, that's Fiona.
Go on, Fiona.
You now. And here also, I bet this is you.
Oh, that's fine. We can do that. Yeah. That's shared.
So basically, these are cancer cells that we grow in the lab on these little flasks and things.
This is what the lady showed us earlier on.
Bits. So ignore these green bits because that's just background.
Right.
So if you watch the video, if it's playing, the cells are growing, and as they turn red, I've put a CRISPR in, so that's going to switch off a gene. So as soon as you see a red cell, that means that there's a gene in there that's been switched off. Every time you see a green cell, that means that cancer cell is dying. So what you can see here is that as the red gets brighter, the cells are all getting green because what the gene I've switched off is essential for these cells to survive. So as soon as I switch it off, they die.
Okay.
So
curing the cancer.
Yeah. So this is obviously just in a lab in a plate like this. So this is a positive control. So I knew this was going to kill the cancer cell before I even started. This is actually a target in the clinic, so we know it works in the clinic. But what I do is use the same technology to switch off genes that we don't know what they do. So we don't know if the cancer cell is dependent on them.
Right, okay.
And I've gone through the entire genome, switched off every gene in turn in over...
How many is that?
Just over 20,000. So, but we grow our cells in these big flasks. I've done it in, well, me and the team, there was about 40 of us involved in the project altogether, have done it in over 300 cancer models, so over 300 of these different cancer cell lines.
I was about to ask, what cancer was that one?
This one's colon cancer.
Okay.
So we've done 30 different cancer subtypes with this technology at a whole genome scale. And the aim is to identify dependencies in every cancer cell, and then that way we can find new targets to develop drugs against. So we can go into the clinic and just target the vulnerabilities of the cancer rather than chemotherapy. The way that works now, it's systematic. It's going to target maybe every cell that divides quickly, which is why cancer patients lose their hair. They've got immune systems are damaged, their guts are damaged, because it just hits every cell that divides quickly.
You're going to try and target just that?
Yeah, so it's all about identifying the vulnerabilities of the cancer cell that the normal healthy cells aren't vulnerable in that way.
And how would you administer that treatment?
So that's the pharma company's problem. I won't have to solve them.
Okay.
But it could be a pill, it could be intravenous.
Right.
I know some of it they'll just inject into muscle in your stomach.
Really?
So depending on the type of compound that they develop that'll decide what way it's administered. For some of the cancers that you have, they try administer, like say esophageal cancer, they might try actually deliver it down, like so that's your food pipe.
Direct.
So they might try get it in like that. So depending on what kind of cancer, some of them are easier access than others. Like you can imagine to get into your esophagus, into your stomach, even into your lungs if you have an aerosol, it's an awful lot easier than maybe getting into the brain and things like that. So where you get your cancer often depends on how it gets treated. Like you're all gents here, you're very lucky. Testicular cancer has a 10-year survival rate of 99%. So 99% of patients who get diagnosed with testicular cancer are going to see 10 years. Whereas with the pancreas, only 1% of patients are going to see 10 years because we just don't know. That's what we call clinical unmet need. We don't know how to treat pancreatic cancer very well.
We don't even know how to diagnose it early enough.
Yeah.
So
That's often the case, isn't it?
Yes. So when people get the diagnosis, even if the cancer itself can be surgically removed, it often already spread. It's too late.
Is it often that it's found because it's spread? Is that typical, or?
I think it can be, but I think for pancreas cancer, just when it grows bigger, it will press on other organs and then just discomfort, the symptoms that would
So
come
with the
with
200 types of cancer that there are, is that it? Or is there more? Is there going to be more like variants
that haven't been found?
I think there are more variants that we don't know of. And knowing of them will allow us to stratify them better and stratify the treatment better.
I want to know. You want to know.
So you said you've done like 300 of these, 300 different types.
300 different models that represent 30 different cancer subtypes.
Right.
So I think for lung cancer, we've done 42 different cell lines. Each of those cell lines will have been derived originally from a different patient. So everything I have here originated in a patient. Some of them like 30, 40 years ago. And we've been growing them in the lab in these flasks and doing our experiments, because we call this in vitro, so it's just in a flask, it's not in vivo, which would be in the body. Because I can't do these experiments in the body. Like I have to start in a flask, and you know, you have to figure out the basics of the cancer in these type of systems. And then that's why we have to sometimes move into a mouse model to check, you know, toxicity of a drug or something before you go into a patient, because a lot of the problems would be off target as well. So I might put a drug on these cancer cells and it'll kill all cancer cells, but I have no idea what's going to happen if it hits a normal
You might
cell.
then kill the mouse.
Yeah, exactly, which happens sometimes.
Patients might die from side effects of the drugs. That's why you have to manage the dose that you give to a patient as well. So a drug can work perfectly on a tumor cell. If the patient cannot tolerate the drug, it is no good.
And I suppose then every person is different too, right?
Oh yeah, exactly. Yeah, like my mama had leukemia when I was 17, and there was probably 11, 12 people went through treatment at the same time as her. She came through it and she survived that time. But she had an incredibly positive attitude. She decided, like she was told at the start, 1 in 5 people survive this type of cancer. And yeah, originally she was shocked, but then she went, well, why can't I be that 1 in 5? And she was an incredibly fit woman as well, worked all her life. So her body was able to cope with it. But at least two of those 11 women were cleared of the disease, and their bodies never ever recovered from the treatment because it was just so hard.
Yeah.
They were
the
so
disease
weak.
that killed them.
It wasn't. Yeah, it was the treatment.
That is the case of my mum.
Yeah.
So she survived the cancer, luckily, but she still suffers from the side effects.
Yeah.
So she's in early retirement at the age of like 55.
Can I ask a personal question? You both had personal relations to cancer. Is that what's driven you to do what you do?
It's what got me into it.
I never thought about it, to be fair, but it might have been.
Yeah.
I mean, I never questioned why I feel so strongly
Just
about
interesting
cancer
that you
research.
both mentioned.
Yeah. Well, for me, it was when I was 17 and we were chatting to the nurses at the time, and the treatment for leukemia at that time had been the same for the previous 30 years. And she said the biggest breakthrough was better anti-nausea drugs to help patients with the side effect. So like 17-year-old me was like, oh, that's not good enough. We have to do better. So I decided to go into cancer research. But now I realise it's actually quite hard. So it's not like there was nobody.
Kicking 17-year-old you.
Yeah. How stupid and naive could you be, kid? But
I think you start adjusting your goals. In the beginning, I was like, oh, I want to cure patients. And now for me, if I can just have one patient live longer for a month, that will have given me enough in my life. As in, that will have been a perfect outcome for me, for that patient, for that family. So it's you can't cure them, not necessarily, but giving them slightly longer life, quality of life, is what we can work towards.
That's brilliant.
Yeah, because my mum, she passed away last year of cancer the second time, and when she was diagnosed like August 2.5 years ago, and they said she probably wouldn't see Christmas, but that they had this trial in America had just finished, and it was quite successful at controlling the disease for a while. So they said, we're going to put you on it, and it controlled it for 2 years. Like, we got an extra 2 years with her, and the problem was she couldn't have chemotherapy because of the first time she'd had chemotherapy, there was damage to her heart, which meant she just wouldn't be able to cope with it again because it's such a hard treatment. But there's a group of scientists somewhere in America who published a paper about 4 months before she got diagnosed recommending this treatment to control the disease, and we got like an extra 2 years because of that study that they did. So exactly like you, I hope that someday something I'll do will just affect some family somewhere. And like my poor mum, she was hopeful to the very end that someone would come along with like a magic pill that she kind of knew it wouldn't happen, but you always have it. But like maybe someday there'll be a family somewhere that because of what we do, a magic pill will come along. So.
That's amazing. I guess it's also like a massive thing with it being able to share all the information.
Oh yeah.
That massive, literally just saying.
Crazy.
Yeah, everything is open. Like it's welcome trust policy that all data gets released quickly.
That makes so much sense. Like, why would it not be? Was that always the case from the start or not?
No, not really. Like science is really competitive. Like our funding would be based on the amount of papers that we get published, and you want to get a paper into a journal that a lot of people are reading. So it's really competitive. Like it's, we don't get.
It shouldn't be. It should be competitive.
It should be collaborative, and that's the way it's changing now. That funding bodies will actually award the biggest grants to international collaborations, and if you can prove you can collaborate well with other people, because that's how we're going to combat it. And somewhere like Sanger as well is fantastic for kind of interdisciplinary stuff. Like we're biologists by training. Like my closest colleague is a computational guy because I generate a ton of data. He does all the analysis. Like without him, my work goes nowhere.
This a minute ago, weren't we? It's like this mental thinking that you can generate so much data. How do you digest it? Like how we were looking at that. I forget the gentleman's name that stood up and spoke. Him putting it into digestible content, I suppose. It's nuts.
Yeah, when I started here, they gave me the data. I was like, here, analyze it.
Have fun.
I was like, open it in Excel, and Excel crashed. I was like, great, I've reached the level where I have to learn bioinformatics. I did, but that is really what it takes. So it really takes people working in the lab, people doing the analysis, clinicians working together with pharma companies as well to bring it to the patient. And we can't do that without communicating with other people. And here they do a really good job. They're communicating between scientists, between the different disciplines.
You all want the same thing, don't you? You know, the end result, you all want the same thing. We all want you to, get a result.
Why are you guys here?
So we're doing a 3,000-mile road trip to raise awareness for Cancer Research UK.
Oh, that's great.
So last year they did the trip.
So last year our friend turned 40. Luke, who unfortunately can't be here today, it's his little boy's birthday. But he wanted to do a Top Gear style journey across Europe. So last year we did that. There was five cars, £500 cars, and do you know what? Because none of us can escape it, we did it for Cancer Research UK. We raised 20 grand.
Oh wow!
20 grand.
Well done,
wow.
Yeah, four days across Europe in £500 cars, raised 20 grand. So this year it was like, wow. So Cancer Research kind of reached out to us saying, like, well done.
Saw what we were doing, didn't
Thank
they?
you. And then this year we've decided to push ourselves a bit more and the cars a bit more. So we're doing 3,100 miles in 5 days. So we're going to Romania in British touring car style cars.
Nice.
But again, £500 cars with £500 to spend on them. 300,000 miles in just an extra day. So we've got the first day is 810 miles, the second day is 800 miles, the third day is 600 miles. So it's going to be brutal.
The last day's a lap.
But we want to try and raise 40 grand. Finish
the cars.
Yeah.
Wow.
So we want to try and raise 40 grand.
12 of us.
That's brilliant.
We're already at 13 and we leave at the end of the month. So coming here and speaking to you guys, like it's incredible.
We're on Instagram.
Yeah.
The cars are finished yet.
We've all got issues. Ours has been wrapped today.
Crush cancer, not cars. It is.
Oh, fantastic! Because that's what I think. Was it you? I was saying that one of our taglines is "Beat the disease, not the patient." So it's kind of this similar bit.
Oh, that's good. That's good.
That's fantastic.
Yeah. So
hence why the YouTube camera.
So Patrick's new with this. Patrick works with me, but he's coming along to document it because then if we want to try and double the money. So if we can get it out and about, then great.
So that talk was incredible. So good camera. Good job, everyone.
So what's brought you here today? We did a charity drive last year, raised quite a lot of money. Cancer Research UK got in contact with us for the money that we raised. I think we targeted 6 grand, raised 20. So we clearly ticked a metric somewhere that allowed us to then be a bit more active with Cancer Research. So we've been invited along today to understand where the money that we raise is put to use. So there's 6 of us here now this evening just to understand that,
So has it been useful hearing from the researchers today?
It's been incredible. My brain hurts. The reason why there's 6 of us, there's 12 of us driving across Europe is because I speak for myself, not those guys. I'm not clever enough to do the research. Listening to the researchers is incredible. I know Patrick spoke about their enthusiasm, but what really shocked me is to make the difference, they're like, if they can just extend one person's life for 1 month and allow their family and their friends to have that loved one around them for that little amount of time extra, then they'd be happy. Whereas I think I'll come here, it's like, well, cool, we're going to drive across Europe and beat cancer. It's nuts, just the small scale but massive wins that everyone's getting. It's crazy.
So that is going to conclude this video. I've learned a hell of a lot. I know you boys have learned a lot as well.
My brain hurts.
Yeah.
How was it for you?
Amazing.
Good?
Yeah.
It's nuts.
Learned so much. If anybody wants to know where their money is going towards, call any of us. It's incredible.
Eye-opening.
Yeah.
And we're going to come back.
Yeah, we'll come back. The team here have been really, really helpful and really supportive. So I know you've got a lot of footage from them probably coming in. We'll have just seen. They've been super helpful. Like they want us to come back with the cars.
If they make it.
When we make it.
Yeah, when we make it.
So we can fuck cancer.
Yep. Yeah. Fuck cancer. Thank you very much for watching. I hope you enjoyed this video. Make sure you liked and subscribe to Talk Nation so you can see all these other videos on the tour. And of course, make sure you hit that link in the description to donate some money to our cause. Follow Crush Cancer Not Cars on Instagram as well. Thanks very much.